Hello, everyone. Today we will talk about lipids. Lipids are a family of substances that are insoluble in water but soluble in nonpolar solvents and solvents of low polarity, such as diethyl ether. Unlike the case with carbohydrates, we define lipids in terms of a property and not in terms of their structure. Lipids play three major roles in human biochemistry. They store energy within fat cells. They are parts of membranes that separate compartments of aqueous solution from each other. And they serve as chemical messengers. An important use for lipids, especially in animals, is the storage of energy. Plants store energy in the form of starch. Animals, including humans, find it more economical to use fats instead. Although our bodies do store some carbohydrates in the form of glycogen for quick energy when we need it, energy stored in the form of fats has much greater importance for us. The reason is simple. The burning of fats produces more than twice as much energy, about 9 kilocalories per gram, as the burning of an equal weight of carbohydrates, which is about 4 kilocalories per gram. The lack of water solubility of lipids is an important property because our body chemistry is so heavily based on water. Most body constituents, including carbohydrates and proteins, are soluble in water. However, the body also needs insoluble compounds for the membranes that separate compartments containing aqueous solutions, whether they are cells or organelles within the cells. Lipids provide these membranes. Their water insolubility derives from the fact that the polar groups they contain are much smaller than their alkane-like or nonpolar portions. These nonpolar portions provide the water repellent or hydrophobic property. Lipids also serve as chemical messengers. Primary messengers, such as steroid hormones, deliver signals from one part of the body to another part. Secondary messengers, such as prostaglandins and thromboxanes, mediate the hormonal response. For purposes of study, we can also classify lipids into four groups, and this is according to structure. Simple lipids, such as fats and waxes, complex lipids, steroids, and prostaglandins, thromboxanes, and leukotrienes. This is a diagram that shows the relationship in structure of the simple lipid and complex lipids. So simple, complex. Simple lipids consist only of glycerol and fatty acids, whereas complex lipids contain additional groups such as phosphate and carbohydrate. So we have glycerophospholipids, which consists of glycerol, fatty acids, a phosphate group, and alcohol. This is sphingolipids. If instead of glycerol, it contains a sphingosine, fatty acid, phosphate, and choline. Now, the glycolipids contain a carbohydrate. So you can have sphingosine, fatty acid, and a carbohydrate. Steroids are another class of lipid molecules identifiable by their structure of four fused rings. One, two, three, four. One, two, three, four. Although they do not resemble the other lipids structurally, steroids are included in lipid category because they are also hydrophobic and insoluble in water. Prostaglandin, thromboxane, leukotriene. Prostaglandin, thromboxanes, and leukotrienes are three classes of arachidonic acid derivative. This is arachidonic acid derivative, collectively called eicosanoids. They have an extraordinary range of biological effects. Let us talk about the glycerides. But first, we consider the reaction of alcohol and carboxylic acid. This is alcohol, and this is carboxylic acid. This is C double bond O OH. In a condensation reaction, water is removed. So OH from the carboxylic acid and H from alcohol, and an ester bond is formed. This is now the ester bond. That same reaction takes place when the alcohol is glycerol and the carboxylic acid is a fatty acid. This is glycerol and the fatty acid. So you have here, this group will form water if there is only one fatty acid esterified to the glycerol. You call it a monoglyceride. Another a fatty acid, if this is again esterified to the glycerol, then you will have a diglyceride. And if you have three fatty acids esterified to the glycerol, then you have a triglycerides. Triglycerides are triesters of glycerol and long-chain carboxylic acid called fatty acids. This structure shows you the triglyceride, also known as triacylglycerol. So this is the glycerol group. These are the fatty acids, and this is the ester linkage. Okay, so one ester linkage, another ester linkage, and another, the third ester linkage. What are fatty acids? These are all unbranched carboxylic acids. Take note, carboxyl group, carboxyl group, carboxyl group, and so on. COOH. And a long hydrocarbon chain. The, the chain could have a size of about 10 to 20 carbons. So lauric acid, how many carbons are there? 12 carbons. Myristic acid, 14 carbons. Palmitic acid, 16 carbons. Stearic acid, 18 carbons. And arachidic acid, 20 carbons. These are saturated fatty acids. because all bonds are saturated, meaning all single bonds, no double bonds. In this table, we have the saturated fatty acids and the unsaturated fatty acids. And take note of their symbol. If lauric acid contains 12 carbons, then it is given a symbol of 12 for the 12 carbons and 0 for no double bond. So since lauric, myristic, palmitic, and stearic are all saturated fatty acids, then the double bond is 0. Now, in the case of oleic acid, it is also 18 carbon, but there is one double bond. So it is given a symbol of 18, 1. And this number here indicates the position of the double bond. For linoleic acid, this one occurs twice. Ayan, may subscript 2. So kaduha ang double bond. So it has two double bonds. Still 18 carbons, but it has now two double bonds in position number 9 and 12. And linolenic acid, three double bonds. Take note of the subscript here, 3, meaning this structure is repeated 3 times. Take note also of the melting point. Okay, and take note of the relationship of the melting point and the size of the carbon chain, the melting point, and the presence of the double bond. These are various ways of drawing structures of organic compounds, which can also be applied to fatty acids. This is the expanded structure or expanded drawing. All bonds are shown, carbon to carbon, carbon to hydrogens. Okay, and all atoms are also shown. In here, only the bonds between carbon to carbon are shown, pero the hydrogen are still shown. The presence of hydrogen atoms are still shown, although carbon to hydrogen bonds are not anymore uh, uh, draw, are not anymore shown. Pero dito, sa skeletal structure, only the carbon to carbon bond is shown. The carbon and hydrogen atoms are not anymore shown, but it is understood. So if you have this to this line, it is understood that there is a carbon uh, at the end of each line. And since this is all single bonds, meaning it is saturated, so it is understood na kada isa dito merong hydrogen, although it is not anymore shown in the drawing. In here, we have the fatty acids in their skeletal representation. So remember, lauric acid is 12 carbons. So kung bilangin natin, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So yung pinaka-12th carbon is the carboxyl carbon. You can do the same with myristic, palmitic, stearic, and arachidic. These are the unsaturated fatty acids in their skeletal representation. Take note of the presence of the double bonds. Oleic acid, one double bond. Linoleic acid, two double bonds. linolenic acid 3 double bonds. In nature, we encounter only the mixed triglycerides, meaning different fatty acids are esterified to the glycerol. A triglyceride that contains one kind of fatty acid, such as triolein, is synthetic, meaning it is prepared in the laboratory. Yung natural na triglyceride contains different fatty acids. So if we examine this triglyceride, this is the glycerol portion. Remember, the glycerol is a 3-carbon alcohol. Then this is the fatty acid, one fatty acid. This is palmitic acid. This is the ester linkage. The whole chain here is palmitic acid. Remember, ilang carbons ang palmitic acid? So, 14 na yan, 15. Ito, ang pang 16th carbon is the carboxyl carbon. Okay. Etong fatty acid na to is oleic acid. Take note of the double bond, one double bond. And this one is stearic acid, 18-0, 16-0, 18-1. Okay, stearic acid, you are no double bond. You cannot see the double bond. No double bond. This is a saturated fatty acid. Ito ang unsaturated. This is a figure of a complex lipid. Okay, this is the glycerol group. This is fatty acid, stearic acid. Another fatty acid, linoleic acid. A phosphate group, and choline. Okay, this is a structure of choline. Take note, ito siya. So it contains choline, a phosphate group, fatty acids, and the glycerol. This particular phospholipid is lecithin.